Hello everyone. I'll be talking about biomedical research in a liberal arts setting, local efforts and global lessons. I'm Irosha NAWARATHNE, professor in chemistry and a director of undergraduate research at Lyon College in Batesville, Arkansas. If you think of Batesville, Arkansas, it's located in rural Arkansas in the United States. This is where we are. So the Lyon College is a very small liberal arts institution. We call it School of Liberal Arts and Sciences. Where I work, the Batesville campus has only about 800 undergraduate students, so it's very small. We also have a dental school in Little Rock area, and another veterinary school is coming up in the similar area. So altogether, 3 of those campuses include the Lyon College. The research I'm going to be talking about today is conducted by all undergraduate students. These are the current lab members of my group, research group. And then there are so many other students involved in the past, more than 60 students within last decade, who work with me and contributed to the work I'm going to be showing you under the drug discovery at Lyon College. And about my journey, I come from Sri Lanka. I had my BS degree in chemistry at the University of Colombo, Sri Lanka. Then I went to Michigan State directly for my PhD from BS in order to pursue bioorganic chemistry PhD, where I work with Taxol, a celebrated anti-cancer agent, and then try to make it a better pharmaceutical using enzymes to modify the Taxol compound. After that, I went into University of Michigan College of Pharmacy to complete a postdoctoral training in medicinal chemistry. With all those enzymatic work, organic work, as well as medicinal chemistry work, I am able to now conduct independent research as a faculty member at Lyon College. So at the University of Michigan, I worked on antibacterials. At Michigan State, as mentioned, I worked on anti-cancer. So in our research lab right now, we combine the two and then do both lung cancer as well as antimicrobial work. So speaking of a liberal arts institution, also a primary undergraduate institution like Lyon, the faculty are primarily interested in teaching and their responsibility is teaching. And we do a lot of high teaching loads. We also have very high commitment to service to the college as well as community. Because of that, being able to spend a whole lot of time in the research labs or doing research is difficult. The reason why I wake up in the morning or maybe go to my research lab or maybe work on a manuscript or a grant proposal is because of my students who are undergraduates. They are my pride and inspiration. These are the current students. I have so many pictures of my past students over the years, and all of them have gone into MDs, MD PhDs, PharmD PhDs, PharmDs, dental school, veterinary school, optometry school, and then several of them have already completed their PhDs in chemistry, biology, environmental science, or sometimes some of them are still in school for completing their degrees. So that's what we get by working with undergraduates in a research lab. We might not make the greatest discoveries. Sometimes we do. Most of the time, it's very rewarding because I see how my students excel from all the experiences, such as going to a conference, presenting at a conference, making a poster, or giving a talk, or even to just hang out with people different from them. That gives them the ability to thrive in what they do after college, and that's what I find as my reward. And I also wanted to provide them all what I can because sometimes or most times they end up coming back and visiting us, and when they do, I want to make sure they have a lab they can call their lab and they can enjoy it. So that's that. And with that being said, my research at Lyon College is under two parts: pedagogical research, because I'm an educator and I like to teach effectively, making the content relevant and interesting to my students, and I also let them do group projects in the teaching labs. And primarily my research is biomedical, and it's both anti-lung cancer and antimicrobial. So I'll talk about more of this, but I will briefly mention, in case you have questions, you can always reach out to me. So relevance, as you know, matter to people in learning. More and more we've been asked the question, what's in it for me, from the students and even from other people, because, if it's not relevant, we are not going to stay motivated. John Keller says, four major conditions need to be met when people want to stay motivated. One is relevance, the others are attention, confidence, and satisfaction. So with the relevance, I usually try to make my organic chemistry classroom, which I do both organic chemistry 1 and 2 at Lyon College. And although it's a very challenging subject for majority of the students around the world, in my classes, they actually find it very enjoyable, although they do complain it's a lot of work. Well, that's what it is. But they do not want to rote memorize, and I do not want them to just memorize organic chemistry. They should understand it in mechanistic way, and then also a more meaningful way. So the efforts I have made is to, one time I am in my teaching labs and the lecture, I always try to incorporate so many relevant examples so that they know why they're learning it. And then most of this pedagogical work, after it's been tested several years and collect data and surveys, I published them with Journal of Chemical Education. So sometimes, like the alkenes may be introduced to them as a plant pigment, which is present in both fruits and vegetables, or the fall foliage, which we have right now in Arkansas in November. Or it could be something that they can know because ethanol, which is the product, is consumed by people. And then sometimes in organic chemistry, because it's challenging, it can be the answer, that's alcohol. I hope my students are not doing that. But anyway, when I'm teaching these alkene reactions and alkene chain reactions, I talk about how certain things are very relevant to us. For example, plastics. These ethylenes make polyethylene, and as you know, polyethylene is in bread bags, plastic films, shampoo bottles, grocery bags, and many more. And each one of these other plastics are also alkene chain reaction products. So with that kind of relevance and bringing everyday material or everyday things they can relate to the classroom before I teach them alkenes and alkene reactions has enhanced their ability to stay motivated and also do very well in the classes. And as I mentioned, Lyon College is small. However, we are mighty because we do produce some of the best students in Arkansas as well as in the United States, ready for professional schools and graduate school. Another thing I tried in organic chemistry classes is to introduce diversity through an organic approach. How I did it is I asked the students to compare social diversity with molecular diversity, given the importance of diversity these days. So when they did that, they went above and beyond, making very, very interesting reflections. Some used organic chemistry, some used chemistry, some did not use chemistry at all. However, the differences and also their creativity is mentioned in this article, and both these articles were highlighted internationally by research outreach for me, making organic chemistry fun, meaningful, and accessible. If you're interested in more information on it, I would really encourage you to go and check these out. They are freely available online. Project-based learning is also something I try out as a pedagogical research opportunity. I haven't published some of this work yet, but I have a whole lot of data I've been collecting because I allow them participation credit and learning from each other. One example I might give you is group exams, group quizzes, things like that. Other than organic chemistry, I also teach analytical chemistry because we only have 3 faculty at Lyon College who are in chemistry, and I have to be the person to cover both organic and analytical chemistry with my background. So it's enjoyable to do project-based learning in those advanced classes, special topic classes, as well as some of these group activities I very much use in my introductory chemistry class whenever I get to teach it. So please reach out to me if you have more questions or if you want to even collaborate on some of this work, because I'll be very excited. Organic chemistry is hard, no doubt about it. Those who study it have all kinds of trouble. However, we also as chemists have all the solutions. So one thing I ask my students to do in labs or for group projects, bring their own ideas, something that's meaningful to them, relevant to them, to do research on. And some ended up being able to work with microplastics in food and water. Some worked on artificial sweeteners, pharmaceuticals, in natural waters, which will indicate the human contaminations as well as heavy metal presence. So most of these projects started in organic or upper level instrumental analysis kind of classes, and then they went above and beyond to finish them as a long-term research project. So sometimes it can be even figuring out the composition of healthy oil, and this I think is relevant to Japan, because I'm sure you eat a lot of rice as well as you use a lot of rice bran oil in your cooking. So please go ahead and look at these articles if you're interested. So I'm not going to go into details of it, but there are some upcoming manuscripts for some of these group projects that ended up being able to publish, because we have really good data after they did them over years as a research project. And then this is artificial sweetener, Splenda, which is 6 times sweeter than sugar, and it has very adverse health implications, if I may, based on the literature. So we worked on developing antibacterial agents from artificial sweeteners. So lastly, I'm going to be talking about biomedical research because that's what I wanted to really introduce here. So biomedical research is funded. All the biomedical research we do in the lab are funded through NIH, National Institute of Health in the United States, as a part of Arkansas INBRI. So Arkansas INBRI has been tremendously a great mentor as well as a funding source for everybody in the Arkansas who are in a primary undergraduate institution. Because if you think about our universities, we do not have a lot of infrastructure to do drug discovery research. I had to develop it through various grant applications. But now it seems like I already have a very established program as well as a very good infrastructure to do drug discovery work at Lyon. Lung cancer is by far the leading cause of cancer death in the US, accounting for about 1 in 5 of all cancer deaths. It is also the most prevalent cancer occurrence as well as death in the whole world. Each year, more people die of lung cancer than of colon, breast, and prostate. See, both males and females have the highest death rates for lung and bronchus cancers. Quinones, I figured, can be used by undergraduates to develop a very nice pharmaceutical because they're very highly available, ubiquitous in nature, and also they have proven ability to act like biological agents. Or in other words, they can be antibacterial, antifungal, anti-cancer. They have all sort of different activities. Quinone-based pharmaceuticals are very well available. Doxorubicin is anti-cancer, while rifamycin S is a broad-spectrum antibacterial, which is part of it is used in tuberculosis treatment as a first-line drug. And all these other ones are the same. There are many more I did not include here, which are commercial pharmaceuticals. So what's the problem with naphthoquinone? It is considered cytotoxic because when it gets to the cells, they do kill both the cancer as well as healthy cells. The problem is they get into many different pathways in the cell and then have no way to stop them. They destroy all healthy and unhealthy. So in order to be able to make potential pharmaceuticals out of them, we have to systematically discover them. And this is the great opportunity for me because I have so many students who are wanting to do this kind of research, and we have all the time in the world to slowly develop them into pharmaceuticals or lead compounds. So with that being said, I don't plan to go into great details, but we have the ability to do certain things to the naphthoquinone cow in order to make them more suitable for pharmaceuticals. Some of the students who pioneered this work are in the pictures given here. And this work is also in review for publication when it comes out. I would really recommend you to go look at it if you're interested. So they use some certain reactions called modified Michael addition and click chemistry, and then develop these new analogues. And these analogues, as well as some other analogues called rifamycin derivatives, are developed in the lab by undergraduate students. And when they do, we test them against lung cancer afterwards. So originally, rifamycin modifications were done not for lung cancer. We actually made them for finding novel therapeutics for multi-drug resistant tuberculosis. However, they're naphthoquinones. They have the middle part, the blue part, naphthoquinone core. Because of that, I figured, well, why don't we try them against cancer? And we did see very good activity without a surprise. And that's why I consider here one stone is the naphthoquinones. Kill the two birds would be both antimicrobial as well as anti-lung cancer. So similar type of reactions enabling and the click chemistry led us to many different derivatives, which are also going to be published with ChemMedChem. And we also have further modifications we can do to all those derivatives by going through another reaction and then making them to benzoxazinorifamycins. Why we do that? Why do we include these extra ring structures? Is because these compounds, this side has become like they are in certain commercially available rifamycin structures, and those molecules apparently happen to have less adverse effects compared to the rifamycin structures. So that's why we are going there to make them better pharmaceuticals. So process of developing one new compound is very, very lengthy. As you can see, you have to start the reaction, you have to work them up, you have to purify them with something called flash column chromatography. Or in rifamycin's case, we actually have to purify them using prep HPLC. And then when you finally isolate the pure compound, that's not it. And also I want to remind you, this type of very modern instrumentation is all bought by grant money coming from Arkansas INBRI, which is a part of National Institute of Health, as I mentioned. So those are the things I had to do in order to get to the infrastructure to be able to do drug discovery at a liberal arts institution. So once we have it isolated, we characterize it fully by mass spectrometry, NMR, UV visible, and Fourier transform, IR, or the infrared spectroscopy. In order to know what we think we made is what it is. So some of this instrumentation we do have in the lab, but acquired by the grants, external grants. Some we don't. We don't have a high field NMR, for example, or a mass spec that can do large molecules like LCMS. What we do is we pack up all those, and then whenever there is a break or a summer, we go to large institutions and run these experiments to get them characterized. Collaboration is the key even to be able to finish the biological profiling of each isolated and characterized compound. Only things we can do at Lyon are the very low biohazard level, like non-pathogenic E. coli. We can test it against our compounds. We can test the compounds against that E. coli. But any other higher hazard level bacteria, we have to outsource it. Same goes for lung cancer. So if you think about lung cancer, which I started with, we do not have any of the lung cancer cell lines or any human or mouse cell lines at Lyon. What we do is we collaborate, we outsource it. We basically collaborate with many people, many wonderful people at University of Arkansas Medical Sciences to get our work going. So with that, I would show you some activities. So here we are looking at EC50, which is called half maximal effective concentration. What that means is if it's 3 micromolar for rifamycin, for example, that actually means at 3 micromolar, it can kill 50% of the lung cancer cells. So lower the better, right here. Those are rifamycins. But in the case of naphthoquinone, the small one, we do want them to be moderately active because, as you know, regular course naphthoquinone or juglone, the other kind of those ones are toxic to the healthy cells as well. So you want to go away from being toxic by modulating the activity. So something like this, 17, 11, those are pretty good options for us to continue. So all this work is against Lewis lung carcinoma, a mouse model. And then if you think of testing them for their toxicity with healthy cells and the cancerous cells, many different kinds of cancer cells, out of these 3 compounds in little naphthoquinones, this one seemed to be the best because if you think about it, it kills cancer cells at very low concentration, but it does not kill healthy cells at even higher concentrations like 30. For example, this one, it seemed to not be very effective, while the top one, INN7, is killing even healthy cells quite a bit. So this is not a good one. And in the case of rifamycins, I'm just showing you two. One is really killing everything and anything, while INN17 is only very useful to kill the cancer cells while healthy cells are thriving. So that's a good lead. So with that, we have a lot of modified compounds. There are rifs and naphthoquinones, like small naphthoquinones. We know they work against anti-lung cancer as anti-lung cancer, but we don't know how they kill it. So in order to understand how they target or what they target in the cells, we do proteomics. How you do this is pretty much you have untreated lung cancer cell and the treated one with our compound, and then we use LCMS, MS or tandem mass to figure out the different amount of proteins available. And then accordingly, if there is a differentiation between the treated and untreated protein mixes, then you can tell, oh, maybe this is the pathway, maybe these are the proteins my compound targeted in the cell to kill the lung cancer cell. So that's kind of how it goes. So basically, here I'm showing you how treated and untreated. So this is treated with a small naphthoquinone. Here it's treated with the rifamycin modification. And then when you have untreated and the treated one, this brown part or the gray part is not changed. The proteins there are the same for treated and untreated. But these ones are upregulated, meaning these proteins are higher in the treated one compared to the non-treated. And here these protein amounts are lower than the one that's not treated. So based on that, I'm just showing you one example where this is down-regulated or have small amount of this certain particular protein compared to the untreated sample. So if I look at it, that's called SPRC in mouse because this was the mouse model. We also have data for human. And when you look at it, you can see it's both down-regulated when it's treated with naphthoquinone or a corresponding rifamycin compared to the untreated. This shows the same thing. And then if you look at what SPRC in mouse is, it's a protein that regulates the cell growth. So you can probably tell, oh, maybe that's why it kills the lung cancer by targeting that specific protein. But is it really? I don't know, because these are very preliminary work and we have to look at all those proteins and then figure out pathways to see whether one is dominating than the other. So can we really tell how it is killing the lung cancer? So this is like pretty preliminary work. But anyway, speaking of rifamycins, they were made as a part of developing antimicrobials for tuberculosis treatment. So I'm not going to go into details of tuberculosis, but I will show you the data for it. I wonder what that is, but sorry about that. But other than that, like here you have the data for antibacterial activity of our compounds, where I'm not going to go into detail, and I really encourage you to look at the paper when it comes out. It's almost ready to go out for to the public. And surely we have a very good antibacterial activity against MRSA, a very virulent methicillin-resistant Staphylococcus aureus version that we work with the collaborator in order to get the data for. So rifamycins do show their very nice antibacterial activity, while naphthoquinones do not. They did not so far show any antibacterial activity against anything we tried. We have tried non-pathogenic E. coli, Staphylococcus mutans, which is a tooth decay bacteria, and then some fungal species as well. I'm not showing all the data, I'm only showing you MRSA data. So with that being said, I think you get the idea of how we do drug discovery research or biomedical research, as well as any other pedagogical research at Lyon College, a liberal arts small institution. For all this work, both pedagogy, development, and the drug discovery with so many undergraduates, sometimes transforming their lives, I have received the 2025 Rising Star Award for Excellence in Scientific Enterprise from the ACS, the American Chemical Society, which happened to be the world's largest chemical society. So with that being said, I am coming from Sri Lanka, and then I survived United States, and I have made this far. So you can always, if you're a student listening to this, I would say, or even an educator, I would say no matter where you are, how small you feel what you do, keep doing it, because that might make a big difference of somebody's life. With that, I think I want to thank all my funding agencies, both Futureful and Arkansas and Bree especially, for developing the infrastructure with all the applications I have done so far for grants, and then Lyon College and many of my collaborators, and especially my students. Please reach out to me using my email. I'm very good at keeping up with email, so if you do email me, I will get back to you. Thank you again for your time. I look forward to talking to you, emailing you, and then also maybe collaborating with you. That would be lovely. Take care.